Pteridophytes, all the vascular seedless plants, which includes clubmosses, Lycophyta, and ferns and horsetails, Pteridophyta. So Lycophyta and Pteridophyta both fall under the group Pteridophytes. These are sporophyte generation dominant plants. So the sporophyte generation is larger and longer lived, and the plant spends more energy and time growing this generation. They have evolved vascular tissue, which includes vascular bundles comprised of xylem and phloem, and most of the pteridophytes have roots. The xylem transports water from the root to the tips of the stems and leaves, providing structural integrity, which allows pteridophytes to grow taller. We also see the development of branching stems, which is indicative of increased growth, and a waxy cuticle that reduces water loss and creates a barrier to the outside world. The gametophyte stage of pteridophytes is small and fragile and sensitive to desiccation or drying out. And a thin film of water is required for sperm to swim from the antheridia of the gametophyte to the archegonia of the gametophyte in order to accomplish fertilization. And this sensitivity to drying out and requirement of water for sperm to travel to the egg is why pteridophytes are still very dependent on water and moist environments. Let's take a closer look at the vascular bundles that allow these plants to grow bigger than the other seedless plants that we've talked about. So in these cross-sectional views of the fern stem and root, on the top left, you can see a cuticle on the outside, followed by an epidermal cell layer just below that, and vascular bundles, 1 in the root and 4 in the stem here. The bundles are comprised of a phloem layer on the outside and a xylem layer on the inside. And the xylem and phloem are specialized tissues for conducting water and nutrients through the plant, found in the vascular bundles of vascular plants. The xylem is mainly woody, meaning that it is comprised of non-living cells. And in ferns, this includes tracheids, which are a vascular type of cell made of lignin, which is a tough, rugged material. And this helps give the plant structure. So you have these lignin-comprised tracheid cells within the xylem that help transport water and water-soluble nutrients from the roots to the rest of the plant, while the phloem is the living material that transports nutrients, especially the sugars from photosynthesis from the leaves to the other parts of the plant. Something important to talk about with pteridophytes is homospory versus heterospory. So ferns have equally sized spores, and this is known as homospory, but clubmosses have 2 different size spores. They have microspores and megaspores, and these are this heterospory is a type of sex differentiation, which where the megaspores will grow into female gametophytes with archegonium and produce eggs, and the microspores will grow into male gametophytes with antheridia to produce sperm. Mega, egg, archegonium, producing eggs, and then microspores have the antheridia, anther, like antler, like on a male deer. Anyways, that might help you remember. But this is thought to be an important evolutionary step towards seed production, which we'll talk about more next week. In the image on the right, you can see how much smaller the microspores are compared to the megaspores. There are also differences in the spore-bearing structures of lycophytes or club mosses and pteridophytes or ferns. So in ferns, the spores are produced by sporangium found inside sori, or these clusters on the underside of the fronds, while in club mosses, these spores are found in specialized structures growing out the tips of the club moss, and these are called strobili. And even though horsetails are more evolutionary similar to ferns, they produce spores on strobili like club mosses. Horsetails are these stalk-like plants, and there are actually some in the rock garden in the courtyard of Rankin, if you ever get a chance to check them out. So talking specifically about club mosses, though, phylum Lycophyta, some in this phylum exhibit heterospory, which we talked about earlier, the microspores and megaspores, and molecular data suggests club mosses are the oldest phylum of vascular plants, with Pteridophyta being the second oldest. The leaves of lycophyte of club mosses are called microphylls, and they have a single unbranched leaf vein, which is their vascular tissue. This is in ferns and seed plants, it's a more complex structure, and it's called a megaphyll, their leaves. And just to think back to our bryophytes, they have leaf-like structures, but they're not considered microphylls or megaphylls because they don't have vascular tissue. Again, to reiterate, the spores are produced on specialized erect reproductive structures growing out of the club moss called strobili, and a club moss is not the same thing as moss. Again, it's closer to ferns. So here we'll talk about the structures of club mosses. You can see microphylls, these small simplified leaves with a single midvein as their vascular tissue, and the strobili found growing erect from the tips of the club moss itself, and in these strobili, microsporangia and megasporangia are housed, and the megasporangia produce the megaspores, and the microsporangia produce the microspores. Okay, we're going to skip life cycles of the club mosses and switch to Pteridophyta, which are ferns and horsetails. These plants exhibit homospory, so their spores are the same size, and again, the Pteridophyta are the second oldest group of vascular plants. The leaves of ferns are called fronds. They're highly divided, and when young, they're called fiddleheads. This is when they're curled up in the ground before they've reached out and matured. Their leaves are considered megaphylls with branched leaf veins, so a more complex vasculature. And if we look at horsetails, they're a really different shape than most plants. They have jointed stems with leaves arranged in whorls, and they have microphylls. And where it says here, possibly reacquired through reduction of megaphylls, maybe there was some evolutionary pressure to have less complex leaf structures. Another interesting fact about these unique plants is that their stems often contain silica, which you can actually feel. They feel different than other plants. But let's talk about the structures of ferns here. So the frond is the whole fern leaf, blade, and stipe, where the blade is the expanded leafy part, and below it is the stipe, or the stalk between the blade and the ground. The rachis is the stalk above the stipe, or where the blade is located, the stalk within the blade. And a pinna is a primary division of the blade, so one of these leaf sections, and then a pinnule is a division of the pinna, which can be divided too, but one of these smaller leaf sections. The rhizome is an underground stem, which is going to attach to new fiddleheads or uncurling fronds, those immature fronds that are growing out. And then you have roots on the bottom. The gametophyte is not pictured here. It's very small. Just to remind you, these are sporophyte dominant plants. And the sporophyte undergoes photosynthesis in ferns because it contains chloroplasts, as well as the gametophyte. They're both green. The spores are located within sporangium, which are contained in clusters in sori underneath the fronds of the ferns. That's where they're released when the conditions are right, and those released spores will eventually grow into the gametophyte, the mature gametophyte. When mature, it is heart-shaped and very small, sensitive to desiccation. Upon sexual maturity, the gametophyte will produce both gametangia, antheridium and archegonium. The archegonium are located near the pit of that heart where it comes together, and the antheridium are located near the base where it comes to a point. And through water, sperm will travel from the antheridia to the archegonia and fertilize the eggs the egg to produce a diploid zygote, which goes through mitosis to grow into the mature sporophyte. The sporophyte grows out of what was formerly the archegonium, directly from the gametophyte tissue. So in the life cycle here, we are starting with our mature sporophyte, which is diploid, and it is going through meiosis to produce these haploid spores and release them into the environment. They are dispersed by wind, and if a spore happens to land in a suitable place, it'll undergo mitosis to grow into a developing gametophyte. This gametophyte continues to undergo mitosis to produce reproductive structures at maturity. The reproductive structures are antheridia in the point of the base of the gametophyte, the point of the heart shape, and archegonia in the pit of the heart, or where the two round parts come together. The sperm develop in the antheridia and the egg in the archegonia. And when water is present, the sperm are able to swim through the water, and only when it's present, using their flagella, to the egg and fertilize it. And upon fertilization, a diploid single-celled zygote is formed in the archegonia, and this begins the sporophyte generation, the diploid generation. After many cycles of mitosis, this single-celled zygote grows into a multicellular embryo, all protected within the archegonium structure. And even though the embryo is the sporophyte, the archegonium is the gametophyte. The diploid embryo continues to develop into a mature sporophyte growing directly out of gametophyte tissue. And upon reaching maturity, the sporophyte forms specialized structure called sporangium, located within the sori on the undersides of the fronds. In the sporangium, spores are produced, and they are the sporophyte tissues must undergo meiosis to reduce the ploidy of these spores from diploid to haploid. The haploid spores are released into the environment and distributed with wind. And again, if they land in a suitable habitat, they will germinate and grow into the gametophyte generation again, which completes our life cycle of ferns. I'm going to draw this life cycle out with you guys now. And as always, we are starting with spores. So the spores of ferns are haploid, and if they land in a suitable environment, they will grow through mitosis, consistent with a lot of our other plant life cycles, into a developing gametophyte. And this is a very small, kind of heart-shaped structure in which the gametophyte will continue mitosis to grow and develop the antheridia and archegonia, or the reproductive structures, and the the archegonia tend to be located near the pit of the heart, where the two round ends come together. And just to reiterate, the egg is located within the archegonia, and this is a part of the mature gametophyte. Looking at our male structures, we have the antheridia, which has the sperm, and this would be also part of the mature gametophyte, just the mature male gametophyte. And as a reminder, gametophyte is so with the help of water, we'll get some raindrops on here, and this water, sperm are able to travel to the egg from the antheridia to the archegonia, and in the egg, fertilization will occur. Fertilization is going to bring our haploid gametes, our haploid sperm and egg, and produce a diploid single-celled zygote. So, within the archegonia, in the pit of the two round edges of our heart-shaped gametophyte arises the sporophyte generation, growing directly out of the gametophyte tissue. This sporophyte is diploid, and eventually will grow into the fern that we recognize, and mature. Upon maturity, the sporophyte will develop spori on the underside of the fronds or the leaves. Within the spori are going to be clusters of sporangia, which contain the spores, and before the spores are released, the sporophyte must go through meiosis to change from diploid tissue to haploid tissue, and in doing so, it is producing the spores that will be distributed through the environment with the wind, and if they land in a suitable location, grow into our heart-shaped gametophyte. And complete the fern life cycle.